Okay, cool. Awesome. So thank you so much again for the invitation. I'm really excited to be here. And to get started, today I'd like to tell you about this project that we are here undertaking at my group at the International Laboratory for Human Genome Research, which is part of UNAM, with the National Autonomous University of Mexico. And this is the title of it, Genomic Profiling of Melanoma in Mexican Patients. Now, I thought it would be interesting to this audience since it has genome sequencing and bioinformatics at its core. So let's get started. Okay, so this is the overview of this talk. So first, I'll give you a brief introduction to what melanoma is, and then I'll tell you about this project specifically, called the Mexican Actal Indigenous Melanoma Project, and then some summary and perspectives. And then finally, if there are some questions, I'll be happy to take them. Okay, so what is melanoma? So melanoma is a type of cancer, specifically a type of skin cancer, and it is the most aggressive dermatological cancer because it represents only about 4% of all skin cancers there are, but in all populations, but causes about 3/4 of the deaths from skin cancer. So as you can see, it is a concern and a growing public health problem around the world. Now, its cell of origin is a melanocyte. So many times cancers are named because of the cell that gives origin to them. And in this case is a melanocyte. So the melanocytes live in the basal layer of the epidermis. So if you see here, I have a picture of a skin. And if you do a zoom in here at the top, then you see the layers, the epidermis, the dermis, and here the melanocytes living at the bottom of the epidermis. And their main biological function is to produce melanin, which is a pigment that is able to absorb the most harmful component of ultraviolet light. So because ultraviolet light is able to cause mutations in DNA and therefore lead to cancer, then melanocytes have this important job producing melanin, absorbing UV light, and protecting the genome of cells around them. Now, the majority of the cases, though, of melanoma are caused by excess UV radiation. So if we do get a lot of sunlight or UV radiation from things like sunbeds and things like that, then the melanin is not enough and to absorb it, and the genome can be damaged. And these can lead to changes in cells and therefore to cancer. And well, it's important also because its incidence in many countries around the world has been increasing for the last 4 years. And you see a graph here. This is from the United States, but for example, in Mexico, something similar can be appreciated, where the National Cancer Institute of Mexico estimates that in the last 30 years, melanoma has doubled its incidence by 500%. Now, the reasons behind it are still discussed in the literature, but I think there's two main causes for this big change. The first one is that we are much better now at detecting these cancers, and therefore that's why it seems that there's more, because people register them more because they diagnose them better, and they're diagnosing thinner and thinner lesions every time as melanoma, so that makes it grow. But also there has been a change in the behavior of in the normal life of humans in the last 40 years, in which cheap holidays to places where there's a lot of sun has become more feasible. And therefore, more sun exposure then leads to more skin cancer. And finally, another important thing is that it is highly refractive to chemotherapy. That means it's resistant to chemotherapy, and that may have to do with the embryonic origin of these cells and melanocytes, and they're also their job, right? So, which is resisting mutations, because they produce melanin, and they're a line of defense in our skin. So as chemotherapy works by inducing DNA mutations and therefore killing the cell, these cells are highly resistant to it. And this is especially important in Latin America, because many patients are still treated with chemotherapy, because other types of treatments are very expensive. So this is important for us here. Now, if we talk about genomes, and this can be done, pretty much is done with any type of cancer, not only melanoma, But here specifically, melanoma tumors can be classified in what we call genomic subtypes, 4 of them. What is a genomic subtype? So basically, researchers can study the genomes of each tumor and find a genomic profile, which means the types and patterns of mutations that they find in these tumors. And then they can find, in this case, for example, 4 different such patterns, right? The first one, here, what you see in this graph, and this is very common in cancer genomic studies, this type of graph, usually called an oncoplot. And what you see here is in each column is a different tumor. So in this publication by the TCGA about 6 years ago, they studied 333 melanoma tumors. And so that's each column, and each row is a different gene, as you see here, the names of the genes. So if you see a colored square, that means that that tumor has a mutation in the gene. So you can see that about 50% or a bit more than the melanoma tumor of these melanoma tumors have mutations in this gene called BRAF. And if you study cell signaling, you know that BRAF is a very important gene for cell growth. Right, so these mutations activate the pathway where BRAF works, which is called the MAP kinase pathway. It's a cell growth pathway, therefore leading to uncontrolled cell growth. Then you have about 30% of the tumors with mutations in the RAS genes and RAS H, RAS K, RAS, which also work upstream of BRAF, so they have the same effect pretty much of uncontrolled cell growth. Then about 15% with mutations in this gene called NF1, which is a negative regulator of this pathway. So they all work in this pathway, as you can see. And in these different ways, then this leads to tumor growth. Now, about 15% of these tumors do not have mutations in any of these genes. So these are what we call triple wild type. Now, an important thing about this kind of analysis is that many times we can find that these different genomic profiles do associate with different patient characteristics and clinical characteristics. For example, the BRAF patients tend to be younger and have a specific other alterations, such as in a gene called MITF, which is important for melanocytes. Those ones that have mutations in the RAS genes have more exacerbated activation of this pathway. The ones with mutations in this gene NF1 tend to be older and have more mutations in their genome. And finally, these triple wild types do not have a UV signature. That means they're probably not caused by UV radiation. We'll talk about that later, and have a more complex genome. Now, before I go to the next slide, I'd like to just point out that this study that was landmark when it was published 6 years ago, studied for the great majority patients from European descent, and from, white backgrounds, pretty much. So the picture that we see in Latin America is a little bit different, and I'll show you in a bit. Okay, now going into a topic, I told you that we were going to talk about acral lentiginous melanoma, or maybe I hadn't told you this before, but now I'm telling you that we study specifically this subtype called acral lentiginous melanoma. So what is that? So I just showed you the classification that we call genomic profile, right? Or genomic subtypes. But obviously, before, clinicians didn't know the genome of the lesion, so they classify them by their morphology and by the growth patterns that they showed. So traditionally, then melanoma has been historically classified in these 5 major subtypes. The first 3 that you see here, called superficial spreading, nodular, and lentigo maligna, are associated to UV irradiation greatly. They occur in parts of the body that are exposed to the sun, and they differ really in their growth patterns. As you can see, this grows more horizontally, this grows more vertically, and this appears in older individuals, and they all also appear in hair-bearing skin. And these are by far the most common in European descent populations. So for example, the populations where the study I just showed you, took place. And then we have 2 rarer subtypes. One is called mucosal, and as its name says, it appears in sites of the body that have, mucous tissue, such as the oral cavity, for example. And this is rarer. And then we have acral lentiginous melanoma, which is a focus of our study. And I'll tell you why in a moment. So this subtype arises in the palms of the hands, under the feet, and under the nails. It's difficult to diagnose because people confuse it with diabetic ulcers, for example, or warts or fungal infections. And it is, as I was telling you about, a rare subtype of melanoma in European descent individuals, comprising only 1 to 8% of diagnosis. However, if we look at a map of the distributions of melanoma subtypes around the world, well, in select countries around the world, then we can see that, for example, here in Mexico or in Peru, the majority of the melanoma cases that are diagnosed are from the subtype acrolentigenous in red. It's also the most common type of melanoma in some countries in Africa and in Asia. So this is an important reason for us to study it, but also there are other reasons. For example, it has lower 5 to 10-year melanoma-specific survival rates than other types of melanomas. You can see here in the survival plot, acral melanoma is in yellow. So this just means that patients die faster when they're diagnosed with this subtype. Also, because it's rare in European descent populations, its causes are unknown. Because it has not been sufficiently studied. So I put this graph because some authors think that one of the causes could be mechanical stress for this type of cancer, because lesions tend to accumulate in the pressure zones of the foot, as you can see here. And this is just one image, but many of them exist in the literature where this pattern emerges. But there are no mechanistic studies yet. So this is an interesting correlation, but we still don't know what there could really be causing this type of cancer, or, for example, if there are any genes that, when mutated, increase the risk. We still don't know that, so it's important to study it. Then it also has a different genomic profile. I showed you before the TCGA study with their oncoplot, and if you see the same graph here for another study with a number of acral melanomas, then you see that these BRAF, RAS, and NF1 mutants are less than 50% of the population of tumors, and more than half do not have mutations in those genes. Therefore, it means that it has a different genomic profile, and there are other different genes that are driving its growth. And finally, because of all this lack of knowledge, there is also a lack of therapeutic options. The best option these patients have at the moment is immunotherapy, but it is really very expensive in Latin America and other low and middle income countries around the world. So it is important, or a motivation of our work as well, to try and identify new potential therapies for this type of cancer. Okay, so I gave you now a bit of a long introduction, but I hope that you've understood why we study this type of cancer, we're interested in it. And now I'm excited to tell you about our efforts specifically to tackle this project. And with this, I'd like to tell you about the Mexican acrolentigenous melanoma project. This has been started at our lab here at LIG in Mexico, in collaboration with other national institutes, such as the National Cancer Institute of Mexico or the National Institute of Genomic Medicine, and other international institutes as well, such as the National Cancer Institute of Brazil and the Wellcome Sanger Institute. And our main question, I would say, we have many questions. We as scientists are curious, and we have many questions that we want to answer. But one of the main ones we have is this one. What is the genomic profile, including driver genes and mutational signatures, characteristics of this type of cancer in Latino populations? And to answer this, well, we have to study tumors from Latino patients. So for that, we started a collaboration with the National Cancer Institute of Mexico and with the Regional Hospital for High Specialty of the Bajío in the city of León to invite patients with this diagnosis to form part of this study. And we started recruiting patients back in 2018. I'll show you very quickly the patient characteristics that we have, at least from the National Cancer Institute, where the great majority of our patients come from. As you saw from the map, all the melanoma diagnoses, about half of them are for this subtype, acral. There's slightly more women than men being diagnosed. Most of the patients come from the south of the country, the southern states, as you can see in this map. That just may be because Mexico City is here and it's just closer to these places. Also, we see that the patients arrive when their tumor is already advanced. So a majority of them have ulcerated tumors, which is the main prognostic factor in this type of cancer. So ulcerated is worse prognosis than unulcerated. Also, many of them come at stage 3, which is, well, a late stage already. And in this database of patients, about 2/3 are still alive, and about 1/3 have died from their cancer. The age of diagnosis tends to be at least, well, it has a massive range, as you can see from here, less than 20 to about 100, but the mean is about 62 years of age. And these are just measurements of how thick the tumor is when it's diagnosed. And these Breslow and Clark measurements, just these specific numbers just means it's actually very thick. So that speaks to its late stage of diagnosis. And the majority of them, as I told you, they happen in the palms of the hands and the sole of the feet. The majority do come from the feet, but there's also some metastasis that these tissues have done to ganglia and brain and, for example, bone and liver. So this is a patient population that we are exploring and inviting to form part of this study. So if the patient agrees and they sign an informed consent, then we ask them to donate a sample of saliva so we can get their constitutional or their germline DNA and tumor material in the form of FFPE, as you can see here in the top, an FFPE block. And then we have to do an extensive manual curation of clinical information from each patient because, well, there's more than 100 clinical covariates reported for these patients. So this has been work undertaken by Christian Molina, who's a postdoc in the lab, and it's very hard work. So I'm very thankful to him for taking that kind of work on. Then when that's done, we proceed to doing DNA and RNA extraction. And after that, we do whole exome and transcriptome sequencing for the tumor normal samples, and we genotype the saliva sample to explore their genomic origins, for example. And we have done that in collaboration both with Sinvestab in Mexico, which is a sequencing center, and also Sanger Institute. So far, we've recruited 226 patients to the study. We think this is one of the largest cohorts that we know of, and definitely the largest of Latino patients, at least that we've heard that is being genomically profiled. About half of them have undergone already whole exome RNA sequencing data. The other half are, being finished very soon, as I understand. But for these 103 patients, we are still undergoing data analysis. We need to do a lot of analysis here to, well, align the reads to the variant calling, copy number calling. We need to make sure that we check for contamination of the sample. We need to check if samples have not been swapped, in their names during the pipeline work, and also remove paraffin sequencing artifacts. So we're still ongoing, but I can show you some results, preliminary results that we have so far. And this is the work of Patricia Basurto, who's a PhD student in the lab. So what we see is a mutational profile of acral melanoma in Mexican patients does indicate that less than 50% of tumors have these classical driver lesions. By this, I mean BRAF and RAS and NF1 that I showed you before. There's a lot of tumors with mutations in KIT, another important driver of cell growth. As well, here you see the percentages, right? 16% BRAF, 14 KIT, and 13% RAS. We also have some samples taken from the same patients. In some cases, it's primaries and metastases, some primaries and recurrences. And so far, we see that the mutational profiles are quite similar, which, well, is expected. But also, in some cases, we see some differences that may indicate some additional lesions that the tumor grows in order to adapt, for example, to a metastatic site. So we're still investigating that, but this is more or less how it looks like from the outside. Also, when we compare that to what has been seen in other populations, we see something similar. So, for example, this study from Australians and this study from the US population, they see similar percentages of tumors mutated with these genes. NF1 is an exception. We see less of that, but the other ones seem quite similar. And we have some potential differences, of course. So a few genes appear in our data set mutated that have not been seen in other data sets. So we're also investigating them as potential new drivers, but this is still early work, so I couldn't say. And well, of course, we'll still need to go and look into these tumors that don't seem to have mutations in any of these genes that are the most mutated in our data set. And we want to try and investigate what drives them. Now, a way to try and investigate, what drives a tumor and is not just mutations is the copy number profile. So, gains and losses of chromosomal regions. So the copy number profile in this data set finds, well, very large chromosomal regions with gains and losses. In the top is our data set, and in the bottom is a data set that has been done with a different technique in a different population 15 years ago. And you see here that these pictures look similar. How did you interpret this? Well, each of these numbers is a chromosome. And in blue, you have gains of gains of that region, and in yellow, you have losses. And the higher the peak, the more proportion of tumors that have that alteration. If we do a zoom in this profile, then we see no oncogenes that are amplified. For example, we see TERT, we see KIT, we see CDK4. A new one that has been described for ACRAL, that's called CRKL. And also we see tumor suppressors that are lost, like such as CDK2 and NF1. There's other regions, as you can see, with peaks here of losses that have so many genes that we still don't know which ones are the drivers. But this is still ongoing work. We're also trying to call mutational signatures, but we don't have enough resolution yet. Mutational signatures are patterns of mutations that can be linked to a potential etiological agent. So, for example, UV light leaves a certain pattern of mutation in the genome. Cigarette smoke, many of the agents, carcinogenic agents in the environment and intrinsic, leave those patterns, and you find them in genomes. Then you can potentially know what caused those mutations, and therefore what caused the cancer. However, we don't have enough mutations yet for this analysis. So with our new data set that's coming soon, we hope to be able to also address this problem. Now, just one last piece of data I'd like to show you. This is for the transcriptome profiling. So this is work of Estefania Vasquez, another PhD student in the lab. And what I want to tell you here is that expression profiles of these tumors, when we compare the primary versus metastases, identifies new candidate genes that could be involved in the tumor spread of these tumors. These genes are the ones that you see in red in this part of the graph, and they belong to processes such as cell matrix interactions, inflammation, cell invasion, and angiogenesis, and some of them are cell surface receptors. So what we're doing now is a CRISPR activation screen for trying to identify which ones of these, because there are a lot, as you can see here, it's more than 600 in this part of the graph, actually can lead to metastatic spread. So that's only starting. This is work that has to be done in mice. So it takes a very long time. so we're doing that. And finally, just we also have this question: if we can find potential therapeutic targets of this subtype of melanoma that I was telling you is doesn't have much therapeutic options. So for this, we teamed up with Patricia Posick, who is at the National Cancer Institute of Brazil, to collect fresh tissue from patients. Because before I showed you it was FFPE, and this is fresh tissue. So half of them from Mexican patients and half of them from Brazilian patients, and inoculate them into immunocompromised mice, allow them to grow and monitor their evolution, study their genomics, and then do tests of different therapeutic agents to see which tumors respond to what therapeutic agent. And of course, it would be the first collection done with patients from our own ancestry for this type of tumor. So we're very excited about this work. And basically, this is the strategy, right? You implant sample in mice from 2 to 5 animals per group. Then you wait until they reach 500 cubic millimeters, and then you can passage into another mice to keep studying the evolution, go to the tumor bank for long-term storage, or snap frozen for short-term analysis, genomic or transcriptomic, for example. So I just want to show you one slide about how this work is ongoing. So these are the patients that, the tumors from the patients that have been inoculated so far. The majority do come from stage 3. So I was telling you before, most of them are Brazilian at the moment, some Mexican, and most of them are primaries. And as you can see, well, when you compare the growth of the tumors in different mice, Many times the different mice show the same growth because it's the same tumor, just implanted different mice, but sometimes they don't, and one of them grows a lot and then some others don't. So that's interesting to investigate if it's a technical issue or if it's real heterogeneity in these tumors is something that we're very interested in. So finally, for the next steps of this project, we want to continue establishing these models until we meet our target of 50 models. We also want to establish cell lines and the histological parameters needed to evaluate this. We want to sequence the DNA and RNA, both of the tumors and of the original tumors and the ones that grew in mice, to compare them and see, what lesions were maintained and what lesions just were lost. And when we have that, then we can start testing the response to potential drugs. And we have some candidates that we want to start testing. So finally, this is my summary. I told you in this presentation that acral longitudinal melanoma is the most common melanoma subtype in Mexico and other countries in Latin America, Asia, and Africa. That its causes, whether genomic or environmental, are mostly unknown, and because of that, there is a lack of therapeutic treatment for these patients. That our project aims to sequence the genomes and transcriptomes of these patients in order to search for genomic drivers and mutational signatures. We hope that establishing these PDX models will allow us to test drugs against potential therapeutic targets. And finally, that we are, with all this work, we're hoping to establish a baseline to describe the characteristics of these tumors in Latino patients. Soon we hope that, well, our findings are actually applicable to other populations. And my last slide, I promise, perspectives. So it's important to keep fostering work and doing relationships within countries in Latin America, as this conference is aiming to do, and I'm very interested in that work. So we want to continue on that line. We want to continue collecting samples from patients in Latin America. We want to combine datasets with other researchers if there are such. We want to form new collaborations to study not only this type of cancer, but also expand to the genomics of cancers that are important in Latin America. And with this, hopefully, contribute to diversify the cancer genomics field, to maximize the usefulness of the datasets that are available to our population, because at the moment, most of them are European descent. And well, I also want to start studies on the genomics of other diseases and types of cancer that are important in our region. And with that, I finish. This is my acknowledgement slide. I am very thankful for everyone that has contributed to this work. It's been the work of many, many people. It takes a village sometimes here in Latin America to get many of these things done. So thank everyone, and I'll be happy to take any questions if there's some time. Thank you.
Yeah, thank you, Dani. We ran a bit out of time for questions, so there are 2 questions, but I will leave you to answer one of them typing, and well, now we have 3, so you will have to answer typing 2 of them. I selected one. Can the genomic profile of acral melanoma change when comparing people with and without albinism? Have you looked into that in our region?
With and without what?
Albinism. Albinism?
Ah, sorry, yes. Oh, well, that is interesting. I have not looked into that. Albinism is, yeah, it's a very interesting condition. I have not looked into that, and I know we're running into time, so I don't want to say a lot of things. But if you're interested in that, please write to me, because it's interesting. It's an interesting area, but I haven't done it. I haven't done it yet.
Okay, thank you, Dani. You can also type the answers in the Q&A. So there are a couple of.
Oh yeah, I will in the chat.
Are interesting. Yes, please. In the Q&A, we have a different panel for that. Please do. And thank you very much for an amazing talk and for teaching us about these diseases in Latin America and how can we approach them. And this was, sorry, your slide for presentation. I didn't put it up before. Forgot. Sorry, girl.